gentlemen, thank you for joining us today. We're going to have a spirited 25-minute conversation about machine learning and its potential to change the drug development game. And both of you are pioneers in this field. I think maybe just to start, it'd be great if each of you gave a quick introduction of your background and what you're doing now and why you're doing that. So maybe Jacob, do you want to start?
Okay, I'll go first. So I'm Jacob. I'm the co-founder and CEO of Iterative. My background is all in chemistry, and so for almost a decade, I was making molecules myself. Go into the lab, put it in the flask, stir it around, analyze the results the next day, and try and figure it out. And so over about a decade, I don't know, I probably personally made 3,000 molecules. I was fortunate; some of them were commercialized in different forms, but we reached an era where miniaturization was coming, automation was coming, the beginning of ML and what's now AI was emergent. But there was this huge dearth of how would you do it for chemistry? As you'll see on Mike's side, biology can make large numbers of things from the get-go, but chemistry can't. Chemists make things and put them in a flask. And so I spent the next about decade building the core technology at the heart of Iterative, which is these little chips. They're not computers, little microarrays the size of a nickel that hold 32 million molecules, and we can measure their interaction with the causes of disease in under 5 minutes. And so in the last year and a half, we've made over 2.5 billion measurements, which is roughly, 10 to 100x the entire history of publicly available chemistry data, which has then allowed us to build AI and ML models on top of that data set that can solve problems that humans never could before. And so we internally use it for autoimmune diseases. And then in partnership with Calico and BMS, we also use the technology in the integrated computation, experimentation paradigm for other diseases.
Great, thanks. Mike?
So unlike Jacob, I'm a reformed investment banker. Was fortunate to cover the pharmaceutical industry during my time in banking, and joined Merck almost 20-something years ago. Spent 18 years working across all parts of Merck. So started doing competitive intelligence, worked on the late-stage pipeline, did the pre-commercialization for a diabetes product, investor relations, business development, ran Merck's vaccine business, spent 6 years in Europe, in Sweden and the UK. And then my last job was running the global product strategy and the strategy for the company, sitting between the research group, the manufacturing group, and the commercial group. And during that journey, a lot of my time and energy was spent on kind of what I thought were the two seminal challenges facing the industry. One was research productivity had been going down for 4 decades in a row. And secondly, but interrelatedly, that led to bad pricing practices and inadequate patient access for medicines. And the only way in my mind that we were going to solve this is if we were able to crack the research productivity question. And about 4 years ago, I came to a very firm belief that this intersection of science and technology would help us understand biology in ways that the human mind alone wouldn't be able to understand it. Despite all of us staring at biology for every day of our life, we really know very little about it. And with the help of machines, I think it's inevitable that we're going to increase that knowledge in a very, very material way. And so I joined Generate 3 years ago. And Generate is a company that was oriented toward how do we understand the interrelationship between protein sequence, protein structure, and protein function. And if proteins drive about 95% of biology. So if I can understand this interrelationship, ultimately it allows you to start to think about how do you program biology with desired effect. And when I first heard about this company, I thought it was science fiction. I had never seen any sort of approach, any remotely similar. Everything had always been artisanal. You immunize a human, a mouse, a llama, you cull through plasma, you see what sticks to a target, and then you try and refine it. This was basically saying, we're going to start with the computer, come up with sequences, build and test all those, and get to answers from a computational foundation. And the early results have been super promising, and we believe that in the very near future, this will be the way all drugs are made, whether it be chemicals in Jacob's case or in biologics in my case.
Got it. So maybe each of you dig a little bit deeper into the actual mechanics of your technology. And Mike, why don't you start first? You alluded to the idea of computational design of proteins. What kind of properties are you able to predict from the outset, and how is that changing the discovery and development timelines that you prosecute?
Yeah, so I think, actually there's a lot of similarity between the approaches that we're both taking, right? I mean, I think, at our heart, we believe at this intersection of computation and experimentation. And, what you need to do that really well, you need some of the best machine learning scientists in the world. We're fortunate, this intersection of machine learning and protein engineering, there were about 90 people when I started at Generate that were skilled at that intersection. These folks are truly unicorns in the world. Our competition for talent for these folks are not life science companies. It's the Googles, the Microsoft Researches. Facebook had a protein team. Those were the competitors. But importantly, it's not just good enough to be a prediction technology. We actually build and test every one of the proteins that our computer suggests. And so we spend a lot of time on some of the technologies that Jacob was alluding to. How do we automate our labs? How do we miniaturize our labs? Spending a lot of time on microfluidics so that we can physically build and test these computational suggestions at a scale that was previously unimaginable. We now will do sets of up to 1 million variants for an individual target. And that all gives us data to refine our computational approach. And so when we started the company, the ratio of public data to proprietary data was 1 to 0. Today, the proprietary data overwhelms the public data set. And that data advantage allows us now to fine-tune parameters from affinity to immunogenicity to potency to a whole range of developability and manufacturability parameters. And if you just think about, across all industries, what computers really are great at is multi-parameter optimization. They are able to solve complex equations in ways that human ingenuity oftentimes stumbles. Getting an optimal therapeutic is a multi-parameter optimization equation. And being able to optimize on all of these different parameters simultaneously gives you a massive speed advantage, but more importantly, it gives you a massive quality advantage, because actually the best molecule is actually what you're really striving for.
Jacob, you alluded to this mind-boggling scale of the experiments that you can do in something the size of a postage stamp. Can you, as best you can to an audience of amateurs, zoom into what's happening in that context and explain to us what kind of interactions you're able to evaluate, and then the relevance of being able to build machine learning models on top of that enormous volume of data?
Yeah, I'd love to. Probably can go for over 16 and a half minutes on the technology alone. It was built in my lab over 6.5 years before we started the company, which is now we're 5 years old. And so it's near and dear to my heart, but also a real transformation for chemistry. And so, biology is able to make these large combinations of things to sort out these problems by combining the amino acids. So in your body, when you get sick with a virus, your body will inherently generate, billions of candidate antibodies, and then basically sample to see which one is good, and then ramp up production. And so, not to speak for Mike's technology, but they've found a way to do that by design for non-natural problems, not the virus. But the issue for chemistry is there was never that Lego-like ability to take things off the shelf and build this large collection of possibilities and test them. And so the chemistry problem is, I guess, similar to the biology problem. Jeff yesterday from Flagship talked about, we've sampled like 1 drop of water out of the entire ocean of chemistry that's out there. And not just 1 drop, but it's like a super biased drop. We put the solutions from things we've done before into the what we know. And so it means that new opportunities that emerge, which are just exponentially increasing with digital data sets on human patient populations, gene sequencing, knockout models, like our correlation and understanding of what causes disease is exploding, but the chemistry solution lag behind. And so our technology is in many ways a way of taking the biologic solution of Lego building blocks, sampling huge collections, finding what works, zooming in and expanding it, and bringing it to chemistry. And so we do that synthetically as chemists now. In the little chip, we make giant combinations of building blocks, millions of combos at a time. We immobilize them into that little, chip the size of a postage stamp. It looks actually kind of like a super tiny muffin tin. And so one molecule goes into each of the little muffin tins. And then we're able to take a cause of disease, like a protein that we know we want to interact with, put it over that chip and measure how every single one of those molecules impacts that cause of disease in under 5 minutes. And the key is that that search space of the ocean is so big, right? It's basically infinite, that you're never going to be able to test everything. And so we use an iterative design, much like Generate, but for chemistry, where we have what we call a diversity collection, where we scout very sparsely but broadly through the solution space. It's kind of like looking for mountains. And so the first piece for us is a foundation model of chemistry, computational, that we call Kawadi, that tells us what's next to what. The next step is this broad diversity search that sort of tells us, like, oh, there might be a mountain over here. There might be one over here. The AI design element then says, like, enrich around that. And so then we come back and within a month build 2 million new molecules, which compared to, as a chemist, we probably made, 100 molecules a month. So the scale of change here is incredible. And so then you map locally, and you're like, oh, the mountain seems like it goes up this way, down that way. And once you have enriched these data sets enough, then the computational multi-parameter optimization Mike talked about can take over and basically, scout the route to the top of the mountain and send you there much more efficiently before. And so for us, it's always from chip to compute, back to chip, and ultimately then to the refined final molecule that will move towards the clinic. And it all comes back to that underpinning, whereas chemists, we're now able to put things together in a manner that biology is used forever.
When you use this metaphor of mountains or spatial proximity to describe the closeness of chemicals to each other, what do you actually mean in terms of closeness?
It's a great question. For us, I think the foundational ability to turn the making of molecules from an artisanal, I don't know if guessing is a fair word, but something close to guessing activity, to a design activity is to represent chemistry as a computational problem. And so that it's not obvious, actually, because chemistry is a drawing of pictures, basically. They happen to be pictures that are all of little hexagons and pentagons. It's like little geometry drawings over and over. But you have to turn that into a math space that the computer can work within to optimize the properties and then take the math answer and turn it back into a drawing. And so actually, in many ways, it's kind of like DALL-E or other AI that we've seen, where you need to take one representation of something and make another representation of it so that you can compute in it and go back and forth. And so Narbe, our CTO, is in the back row there, and his team have built Kawadi, which was recently published, which takes those structures and the math space and puts the molecules into math. And so by next to, what we really mean is that the structure of one molecule is similar to the one that the algorithm would sort of move to next to it. In the mountain analogy, it would be like, you need to know that Arizona is next to California before you go scouting. If your initial model thinks it's next to Massachusetts, it's never going to work. And so we put a lot of work into it, and it actually takes a lot of compute and contrastive learning to then build that map of, like, this molecule is very similar to this one, is very similar to this one, not by having a chemist look at it, but in this smooth, continuous math space that you can then use AI design within and come back and generate back out the actual structure you want to make.
So you both have been involved in building these beautiful molecule generators, and that widens the top of the funnel and potentially makes the candidate molecules that are entering the top of the drug development funnel intrinsically more promising. But so much of the cost, of course, sits in what happens after that, in taking medicines into human clinical trials, which of course is the very expensive, very time-consuming part of the process, and which has always had an incredibly high failure rate. With small molecules, my best understanding is between 5 and 10% probability of success. Historically, biologics, maybe you get twice that, but still pretty bad batting odds. So how do you try to de-risk these molecules thinking through to the development
Yeah.
process, and you know, how much incremental value, when you're talking to investors, are you really able to attach to the quality of those compounds that go in the top of the funnel, when most of the risk, at least from a capital standpoint, is going to be in the clinical stage?
So maybe I'll start and then pass it over to Jacob. But I think when we look at it, the fundamental observation that I think Jacob started alluding to of migrating from this artisanal craft to an engineering endeavor is the transformation we're talking about, right? Increasing predictability. And with our technology, we've consciously chosen certain disease areas where you have better predictability. So if you think about infectious disease, we know very well if you neutralize a virus preclinically, it translates exceptionally well in the clinical domain. We also know we've started to understand the genetic drivers of cancer at a foundational level. So if we can attack one of those genetic drivers with these proteins, the probabilities do change. So we've worked on infectious disease, oncology, and immunology to start to change those odds. We've also have a technology that allows us to dial out biological risk. Part of the IP estate for the traditional pharmaceutical industry is based on the binding region of a protein. So you patent the 6 CDR loops, which is how the protein sticks to the target. Historically, using traditional discovery technologies, you can only change about 10% of the binding region before mathematically you'd expect no functional variants in your library. This technology allows you to change 50, 60%. And because you patent sequence homology claims, ultimately you're able to get around the IP of the existing industry. So we can actually go after things that have proven proof of concept with a better answer outside of the patent scope.
In other words, you're able to change the molecule while preserving its function more than people thought was possible?
Yeah. Okay. Improving it massively, right? Okay. So, a very tangible example, the first program or second program that entered the clinic for us is an asthma program. The precedent molecule is a very good protein. Our version of it is 20-fold better in terms of affinity, 5-fold better in terms of potency. That has massive therapeutic advantages. It'll take the dosing window at a minimum from every 4 weeks to every 6 months. And we have to obviously wait and see what the efficacy differential is, but with 5-fold greater potency, there's reason to believe you could actually have a better therapeutic effect. And so that is a beautiful opportunity for us because you take low biology risk. We know if I bind that target at that location, I've got proven human outcomes, but I can do so with a better answer.
So that de-risking, just to be clear, is coming from your strategic choices. You're pointing your technology at areas where you're able to minimize the risk that's exogenous to the compounds themselves.
Yeah.
Jake, when you think about doing this in small molecule space, is there anything that the machine learning offers in terms of de-risking clinical development consequences?
Yeah, absolutely. It's a great question. Ours is an industry that's incredible because it's a mission-driven one where you bring benefit to patients in need. Also a daunting one because you got to prove it. You got to test it in humans. It's got to work. And so it does present, I think, like Mike, a lot of it, the revolution, sort of must be proven. And to prove it in a business efficient sense, you want to start certainly as a new biotech, new pharma company in areas where the odds are in your favor. And then over time, bring the revolution sort of broadly to all of the different therapeutic areas. And so for us, I would say the biggest impact that our experimentation, computation, ML, AI design piece brings is that a vast majority of small molecule drugs suffer because of their selectivity profile. So typically, when you're looking for small molecule drugs, you have some problem you want to deal with, and you go hunting for some molecule that hopefully sticks to it, hopefully adjusts it in some way. And in many cases, it's really hard to even find a starting point. And then you start optimizing it. And sort of in like phase 3 of the discovery process, you'll then go check and see how selective it is. Not only do I do what I want in the body, but do I not do bad things? Do I not impact the liver? Do I not impact the heart? Do I not impact the thousands of other proteins in the body that it's probably a bad idea to mess with? And so one of the things that's so unique about the Terray approach is that we screen every single target we see against tens of millions of compounds in our library from day one. And so we have this incredible sort of proteome to genome map, this huge like selectivity atlas that now spans hundreds of targets, tens of millions of molecules, which means that as the ML algorithm designs the molecules for progression into the clinic, we start with selectivity from day one that's incredibly different. It's like the whole process, got reversed. And so you start with incredibly selective, safer medicines, from day one. And so we feel very strongly based on our early data that this will lead to better, because to your point, the overwhelming, financial and human benefit in our industry is better. Faster and cheaper is very good, but you got to turn it into better. Like that success rate has to go up. And so we think that because we start with safer, more selective molecules for day one, and the AI can see this entire space and know from the beginning how to avoid those problems, we'll be much more successful. And we've seen early data points. Of course, it will take a while to be all the way through the clinic and read it all out, but we've seen obviously buy-in from extra partners and progression against really hard targets. We've seen incredibly different selective compounds for problems that have been uncracked for years, get done in the last year and a half. And so you can feel that excitement and that incredible transformation in the pace of discovery and the quality. But I think for us, it starts with that selectivity piece.
There are great investors here at this conference, hopefully myself among them. And I want to get advice from both of you. It's a little unconventional, but I want to ask you to imagine yourselves in the seat of an investor and imagine that your two companies don't exist in the market. How should investors who want to be a part of this potential machine learning revolution in drug discovery and drug development pursue that? What do you think is overhyped? What do you think is underhyped? Where do you think there's real value today? And where are we in the overall timeline of this materializing in a meaningful way in the markets?
It's a great question. It'd be so sad if Terray and Generate didn't exist. But
Me too.
You wouldn't get to see Mike and I on our speaking tour with such different design that reflect our backgrounds. But I bet we're going to give the same answer, which is you have to invest at the intersection of experimentation and computation. You have to be able to own your own data, have a platform like a hardware revolution that generates a data revolution that lets the compute revolution happen. If you try and do it on public data, do it with like CROs that you send stuff out to occasionally, you're never going to get there. The key to models that have changed our lives is iteration and feedback. And so you got to look for data that is very large, very high quality, and that iterates quickly enough that it can train those models up. And I think you'll find that in different areas, biologics, chemistry, sequencing, and discovery correlation with clinical data, clinical trial design, whatnot. But it's always the same thing: big, high quality, iterative data that pairs with leading edge compute.
Yeah, I personally would not join any organization in this space that doesn't have an ML orientation. I mean, unless you were to bet on a single asset succeeding, which to me is akin to playing the lottery. I think all platform companies will have to have a digital and ML foundation in the future in biology. You're going to have greater insights than you would using just the traditional artisanal process. I think at the same time, it is not a purely computational endeavor. So you need to be able to complement that strong computation and experimentation foundation that Jacob talked about with people who are savvy in drug discovery and development. Because the regulatory process today does not allow the predictive nature of these sort of technologies and doesn't embrace them. That will change over the next decade, but we're in the very early innings of this. I like to tell my team, we have an advantage because we're the first in biologics. I believe every biologic will be made this way, probably within 3 years, or should be made this way within 3 years. It won't probably happen for a decade. But the reality of it is, when you step back and you think about this, just because we're a first mover doesn't mean we'll be the disproportionate winner. This space, if you're going to be serious in this industry, you're going to have to do this. And just like we saw in the search space, Google didn't invent search, but they are the disproportionate winner. Who wins here is still very early to say. Data will be a big thing. People will be a big thing to determine who's ultimately the most successful. So I think there's a huge opportunity, but I would be very leery of companies that either don't embrace a computational foundation or don't embrace this intersection of computation and experimentation.
Mike, Jacob, thank you both so much for your time this morning, and we'll leave it there.
Thanks for the wonderful conversation.
Appreciate it.